Good morning. We had a bit of a scuffle with the computers. This is a computer number 4, and Dr. Yester saved the day. So thank you, Dr. Yester. That's incredible. So thanks for coming. It is my great honor and privilege to introduce today's visiting speaker. Thank you, sir. Dr. George Mashur. Dr. Mashur is visiting us from the University of Michigan, where he's been for the past decade or so. And this is about how long George and I have known each other, sitting on different committees and different advisory boards. Dr. Mashur did his undergrad in philosophy and then decided that he would like to be a physician. So he went to Georgetown University, where he completed his MD, PhD studies. After that, he went to Mass General, Harvard School of Medicine, to do his residency in anesthesiology, where he served as a chief resident. When he was done with his residency, he actually, as a Fulbright fellow, went to Germany and spent some time in Berlin doing his research before he joined the faculty at the University of Michigan, where he did his fellowship in neuroanesthesia. He rose through the ranks there, and as you can see, he's an endowed professor, Bert Ledoux professor at the University of Michigan. Dr. Mashur is a practicing anesthesiologist who is NIH-funded neuroscientist. He's internationally recognized for the work that he's done in consciousness, mechanism of anesthesia-induced unconsciousness, and very important to all of us, intraoperative awareness. Mashur has authored over 150 publications. He edited 5 anesthesia books. This is how I got to know him first, because I was invited to submit a chapter in his book. He also published some landmark articles I'm sure you've heard about in New England Journal of Medicine, Lancet, and Proceedings of the National Academy of Sciences. At the departmental level, as you know, Dr. Mashur, and you can see on the slide, has been very, very active in multiple leadership functions. He's a senior associate chair for anesthesiology research. At the medical school level, he serves as the associate dean for clinical and translational research and is a founding director of the Center for Consciousness Science. At the institutional level, he serves as the director of the Michigan Institute for Clinical and Health Research, an NIH-funded institute dedicated to advancing translational research. This is a very well-funded endeavor with over $10 million in the NIH funding. So, needless to say, aside from being very active at many administrative levels, Dr. Mashur is an incredibly good clinical or clinician scientist. Nationally, Dr. Mashur serves on the NIH study section. This is when we work together quite a bit, because I was on the SIT study section with him, and he's an editor of anesthesiology and, of course, on the executive board of SNAC, FAIR, AUA, and International Anesthesia Research Society. So it is a great pleasure to welcome him to our grand rounds. I just want to remind you that tomorrow we have our 3rd annual Research Celebration Day. Dr. Quillilan, who is the organizer, put a lot of time and effort into that. We've had a really nice response with over 40 posters that will be presented tomorrow. It starts at noon, goes on till 5:30 or 6. So it's a unique opportunity. Dr. Mashur will have his keynotes lecture at 4 o'clock. So we are hoping that many of you can take time and make time to swing by and hear him talk and get to know him. So today, grand rounds, tomorrow, a Research Celebration Day. So without further ado, Dr. George Mashur.
Thank you very much, Vesna. Thank you for the invitation to speak. Thank you for the kind introduction. Today I'm going to be talking about a unique drug, ketamine, and its effects on consciousness. And as Vesna noted, I founded the University of Michigan Center for Consciousness Science, in 2014, and we study consciousness and anesthetic-induced unconsciousness across the translational spectrum. Computational brain models, doing network analysis, work in rodents, non-human primates, healthy human volunteers, and surgical patients, both observational studies as well as large-scale clinical trials. And I'm going to be talking about some data from healthy volunteers, surgical patients, and also just touch on a little clinically relevant preclinical science as well. Now, when it comes to the drug ketamine, not only have I studied this across all these different models and come to understand it neurobiologically, I've also been able to understand it phenomenologically. So that's me after an MRI experiment. So this cap is an EEG. It looks like I just got out of a synchronized swimming lesson or something. But that big smile on my face is likely because I had just experienced antidepressant doses of ketamine while being imaged. And the individual that I'm standing next to is Dr. Edward Domino, who's now 94 years old. He's still active. He's still funded in publishing. He's a professor emeritus. But a little over 50 years ago, Ed Domino and Gunter Korsen, who's an anesthesiologist in our department, Ed's a clinical pharmacologist, actually introduced ketamine into clinical practice. So that story of ketamine as a phencyclidine derivative was rooted in the University of Michigan. And that was the original paper with that term dissociative anesthesia, which was actually coined by Ed Domino's wife. When he got home and was describing this strange anesthetic state, she said, well, it sounds like it's a kind of dissociative anesthetic. And we'll be talking a little bit about that later. So why study ketamine? It's really a kind of black sheep in the family of general anesthetics, and that's true on a number of different levels, and I'm going to talk about that. So on the one hand, we might be tempted to marginalize it and say, look, it's just a very, very different kind of drug. But I and some of my colleagues felt like, well, if we could understand a commonality between ketamine and drugs like propofol or the halogenated ethers, maybe we could develop insight into a more foundational mechanism of anesthetic-induced unconsciousness. So I'm going to talk a little bit about some of the science to begin with, and then I'm going to get into some clinical and translational research that can actually inform your practice today or help you understand what's going on in the mind of somebody who's had ketamine. So, again, a very different drug on a number of different levels. And roughly speaking, and I think this is a helpful framework for trainees as well, you can group general anesthetics into three basic categories. The first group would be drugs like propofol or etomidate or barbiturates that are more or less selective for the GABA-A receptor. Group 3 drugs, and this is arbitrary numbering, Group 3 would include the halogenated ethers, sevoflurane, isoflurane, and desflurane, which also potentiate GABA transmission, but have a wide variety of molecular targets. As Rod Eckenhoff called it, they're promiscuous in their binding patterns. But there are a lot of similarities between these two groups. And then you have this oddball group of drugs like ketamine, nitrous oxide, and your chair, Vesna, did foundational work in understanding the mechanism of nitrous oxide, I believe in the mid-1990s in a Nature Medicine paper. Xenon, these are sort of the weird drugs that don't quite conform, and I want to show you how they don't conform. So if you think about the molecular target, and I've already alluded to this, drugs like propofol and sevoflurane, which we've studied, that's why I have it up here, potentiate GABA transmission. So they're ramping up inhibitory processes in the brain. Ketamine is not thought to act at GABA receptors as a primary therapeutic target. And if we move from a molecular target to a neural target, such as the thalamus, which is very important for transmitting sensory information and arousal and coordinating cortical communication, virtually every sedative hypnotic depresses thalamic metabolism. Ketamine doesn't. Ketamine maintains oxygen metabolism in the thalamus, and it actually increases glucose metabolism. Now, if we move from this one neural target to a network of neural areas, and in particular a sleep-wake network, there's a special area in the hypothalamus called the ventrolateral preoptic nucleus. And it's special because it turns on when we turn off and go to sleep. Its activation is sleep promoting. And so when that was discovered, it became a very attractive candidate for general anesthetic mechanisms. Maybe anesthetics hijack or co-opt this endogenous machinery for making us go to sleep. And indeed, drugs like propofol and the halogenated ethers activate the sleep-promoting region in the brain. But ketamine doesn't. Ketamine suppresses the sleep-promoting region, and it activates a number of areas in the brainstem and the hypothalamus that normally promote wakefulness and arousal. Now, if we move to the neurophysiologic level, a more global level, drugs like propofol and sevoflurane at surgical anesthetic levels induce a very strong alpha oscillation. So alpha is around 10 hertz or 10 cycles per second in the frontal cortex. It's a characteristic feature. But again, ketamine doesn't. It suppresses alpha and it enhances fast activity in the brain, which is why our currently available awareness monitors or depth of anesthesia monitors, such as the bispectral index or SEDline, they don't really register ketamine. It looks like the patient's waking up, even though they might have a deeper anesthetic. So I was coming at this question of ketamine and its similarities to these other drugs from the perspective of consciousness science. And in consciousness science, it is thought that at a level of information processing, so kind of the highest level, that feedback from the front of the brain to the back of the brain is associated with conscious experience. And so we started investigating this to see what the effects were of different anesthetics on this process that seemed to be important for consciousness. And I'm just going to summarize and give you a schematic of what we found, we and others have found with propofol and sevoflurane. So these arrows represent a measure of directional connectivity, which is a kind of surrogate for understanding how one area of the brain influences another. So in this case, in the awake human with eyes closed, if you're using EEG, there's a kind of dominance of this feedback connectivity from the frontal to the parietal cortex. This straight line represents functional connectivity. And functional connectivity is just the covariation, the statistical covariation between two areas of the brain. So activity in one area is going up and activity in the other area is going up. You can measure that and say that they're functionally connected. And so what happens with propofol or sevoflurane anesthesia is that there is a selective loss of this feedback connectivity. Feed forward from the parietal to frontal lobe is maintained, and the functional connectivity between these areas is depressed. This has been shown with EEG, fMRI, magnetoencephalography. So we had studied this in a number of different ways and said, what's going to happen with ketamine? So with our colleagues in South Korea, we studied 30 surgical patients who had a bolus, well, it was given over 2 minutes, but an anesthetic dose of ketamine, 2 mg per kg, no pre-medication, low-resolution EEG in the frontal parietal area, and then we applied some measures of directed connectivity, again, trying to figure out the influence of one area of the brain on another. And this is what we found. So let me break this down for you. You've got 2 studies here, 2 different measures of directional connectivity from the same data set. Now remember that arrow going from the front to the back of the brain? That's shown here. So these red tracings are the feedback connectivity, frontal to parietal. Blue is feed forward, parietal to frontal. The blue area here that's shaded is when the drug was being given, and everything afterwards is ketamine anesthesia. So what you can see is that as the ketamine's getting infused, that feedback connectivity is dropping, and then it kind of gets neutralized with the feed forward, and that's shown here in the asymmetry between those 2 directions. So the measure here was called symbolic transfer entropy. That's not so important for this morning's talk. This is a different measure called directed phase lag index. And now you're looking at a schematic of the brain that's top down. So this is the front, this is the back. And these arrows are quantitatively informed by the measure. And again, you can see this kind of feedback dominance. So it's very consistent with what we had shown before. It's consistent with this measure. And once ketamine is given, that feedback dominance goes away. Doesn't mean that everything's getting wiped out, but what it means is that net directionality is lost. So, some argued, well, you're giving this big bolus. How do we know that this doesn't happen at sub-anesthetic levels as well? Is this really particular to general anesthesia? So we studied 10 healthy volunteers, and we gave them 2 doses. We gave them the antidepressant dose. That's the one that I had when I had that big grin on my face, about 0.5 mg per kg over 40 minutes. So it's very, very low, but you definitely can sense the dissociative effect. And then we followed it with a 1.5 mg per kg bolus. So same total was 2 mg per kg. This time we used high-density EEG, 128 channels, and some measures of functional and directed connectivity. And I'm just going to show you one slide from this study. What you're looking at here is called a connectogram. So it's a depiction of this functional connection in the brain, and you can kind of understand what this is saying by looking at this schematic here. So this is a bird's eye view. There's the nose, there are the ears. And again, in the baseline and at sub-anesthetic levels, you see this feedback dominance. So very consistent with what we saw. That disappears at anesthetic doses, and then it reappears with recovery. And this was specifically in the alpha bandwidth. So you can see at around 10 hertz, this connectivity is maintained during the sub-anesthetic infusion. Then it disappears. So red is hot, means there's more of it. Blue is cool, but there's less of it. And then it just starts to reappear as these individuals recover. So it appears that this is specific to the anesthetized state. So in asking the question, is at the information theory level or consciousness level, is ketamine, again, going to be the problem child that doesn't really fit in? Well, in fact, it did. And to my knowledge, this was the first neural correlate of general anesthesia that crossed both these traditional GABAergic drugs as well as ketamine. And just to give you a view of what that looks like, this is just a different way of representing it. Again, the red is the feedback, the blue is the feed forward, the gray shaded area is where surgical patients are getting ketamine, propofol, or sevoflurane, and you can see what is qualitatively very similar behavior across all 3 of these drugs, representing 3 different classes on a neural process that's thought to be critical for conscious experience. Now, I don't want to say that ketamine is exactly like the other drugs. There are some critical differences, and I just want to probe a couple of those differences scientifically before moving to the clinical part of this talk that can help inform your practice. So we are interested in this arousal-promoting effect of ketamine, and it had been shown that different areas in the brainstem and the hypothalamus got more active when animals were given ketamine, these arousal-promoting areas. But we wanted to see what was going on in the cortex. Are there increases in arousal-promoting neurotransmitters, such as acetylcholine? So we took 11 rodents, and we gave ketamine. Now, this is going to sound like a crazy high dose, 150 milligrams per kilogram, but that's going intraperitoneal. So the dosing with rodents is a little bit different. We used EEG, and additionally, which we can't do in humans, of course, we put a very tiny catheter into the prefrontal cortex so we could measure acetylcholine levels and assess cholinergic tone. And then we used some measures of functional connectivity. Now, this is another connectogram I've shown you before. The red is hot. It means there's more of this functional connection. The blue is cool. This is the frequency over here, and 10 hertz is that alpha band, and 150 hertz is this higher frequency gamma activity. So LORR and RORR stand for loss of writing reflex and recovery of writing reflex. So how do we know an animal is anesthetized? We can't ask it to squeeze our hands. So rodents like to be on all fours. If you turn them on their back, they're exposed, so they immediately try to get back on all fours. It's called the writing reflex. And it's just common in animal research with general anesthesia that when animals lose that and they're just splayed out and they don't really care anymore, we say, all right, they're unconscious. So during unconsciousness, right here, you can see all this blue. Forget about these lines. That's just electrical artifact. This blue means there's a loss of functional connectivity. Very consistent with what we've shown in humans. Now, I want to point out something that's more interesting as you start to approach recovery and you're getting more and more towards sub-anesthetic levels of ketamine. You can see this screaming red hot coherence in this high-frequency oscillation, which is thought to be important for conscious experience. And what's going on during these times is that you actually see a profound increase in acetylcholine levels in the cortex. So first of all, this is the waking state. And we've done this and published it, if you give animals propofol or sevoflurane, acetylcholine levels drop, just like they do in our brains when we go into non-REM sleep. If you give ketamine, it actually goes up, which is similar to what happens in our brains during REM sleep. And we'll talk a little bit about that connection in a bit. But even more interesting, as you get to the recovery stage, there is a fourfold increase in acetylcholine in association with this high functional connectivity in the higher bandwidths. Now, I mentioned this connection to REM. Now, when you go off to the operating room today and you're giving propofol or sevoflurane or iso, you're probably inducing a state that has some similarities. It's not the same state, but similar traits as non-REM sleep. Low conscious content, slow wave activity, low acetylcholine in the cortex. But when we give ketamine, we're inducing something that's a little bit more like REM sleep. And the characteristics of REM are an active cortex, high conscious content, we're dreaming. We're not conscious of the environment, but we're richly conscious in terms of a disconnected state of experience, and high acetylcholine. In fact, acetylcholine in the cortex and the basal forebrain, which generates it, can go even higher than in the waking state. So we saw this, and we saw this coherence, and we said, what if we kind of engineer an anesthetic to look more like our sleep? So when you go to sleep at night, you first start off more in slow wave sleep, and then as the night goes on, you have more bouts and longer bouts of REM sleep. And it's been suggested that that sleep organization helps prepare the brain for waking up in the morning with this high cholinergic content and activity. So we wanted to see whether or not we could add ketamine to an isoflurane anesthetic and induce something that would be similar to like REM sleep and have both a slow wave sleep-like state and a REM sleep-like state and see what happened to recovery. So we took 10 rodents, we gave ketamine. Now remember, to get a rodent really anesthetized through an IP injection, you need 150 mg per kg, so this is much, much lower. And we did this during 1 MAC of isoflurane. Again, EEG, prefrontal cortex microdialysis, so we could look at the cholinergic tone, and then a measure of directed connectivity. Again, trying to figure out the influence of one brain area on another. So the first thing we saw is that the animals that got ketamine went deeper in terms of the anesthetic. And we knew that because they went into burst suppression. And this is a measure of burst suppression ratio. So the higher this is, the longer the periods are of suppression for a given time epoch. And you can see in red, the animals that got ketamine, after they got ketamine, had an increase in the suppression ratio. So they're getting deeper. I said, duh, of course, I should have known that, right? We're giving more anesthetic, they should get deeper. Why would I think that they were going to get lighter or their brain's going to start to wake up? And you don't need to worry about this, but this is just different points of the isoflurane anesthetic. So this didn't really confirm my hypothesis. I thought the brain was going to get more active and not go into a deeper anesthetized state. But then something interesting happened. Even though the animals went deeper who got the ketamine, they emerged around 45% faster. So they got deeper and they came out quicker. Now normally, if you have an anesthetic and you're giving a neuromodulatory drug, you're either going to lighten the anesthetic and make them come out quicker, or deepen the anesthetic and make them come out slower. So if you give amphetamine, you're going to lighten them up and they're going to come out quicker. If you give another anesthetic, let's say a bolus of propofol, you're going to deepen the anesthetic and it will probably take them longer to wake up. What happened here was paradoxical because the animals got deeper, but they came out faster. And that accelerated recovery in the ketamine group, and this is just emergence time in seconds, was associated with increased cholinergic tone. So here's the waking state. These are acetylcholine levels. Here are the samples taken during isoflurane anesthesia. The animals got an injection of either saline or ketamine, and you can see acetylcholine doesn't really change. So that isoflurane is dominating the brain at that point. It's not like when you're just giving ketamine alone. But once the isoflurane is discontinued, suddenly you start to see the divergence of the ketamine group in terms of enhanced cholinergic tone. And tomorrow in the research lecture, I'm going to talk about how that might actually accelerate the recovery of consciousness. And we also showed enhanced communication across the brain. So we kind of normalized all the different emergence times. Of course, they're happening at different times, but we align them. And this is that feedback connectivity. This is the feed-forward connectivity. And you can see how the red starts to climb up with a higher slope in the animals that were treated with ketamine, suggesting that the higher cholinergic tone and the higher functional connectivity helped get these animals out of the anesthetized state quicker. Okay, you survived the science. Now let's talk about how the effects of ketamine translate to postoperative outcomes, because that preclinical study I just showed you is not unlike things we do in the operating room. Patients on an isoflurane anesthetic, and we give ketamine oftentimes for analgesic purposes. And this was a study that was co-conducted with my close collaborator and great friend Michael Avedon and published in The Lancet about a year ago. So, to assess the effects of ketamine on neurocognitive outcomes of surgery, we studied 672 surgical patients, 65 or older, so it's an older population. They were undergoing major cardiac or non-cardiac surgery. It was multi-center, it was international, and there were 3 arms of the trial. Ketamine, either half mg per kg or 1 mg per kg, bolus dose in between induction and incision. Not uncommon for clinical practice. And then we assessed neurocognitive outcomes for 3 days. The primary outcome was delirium. And the reason why we were studying this for delirium is because there was a relatively small trial of cardiac surgical patients demonstrating that a subanesthetic dose of ketamine during the cardiac anesthetic actually reduced delirium by 10-fold and reduced cognitive dysfunction, and did so possibly through anti-inflammatory mechanisms. It's thought that neuroinflammation is important for postoperative delirium. Now, that's counterintuitive, that you give ketamine and you reduce delirium. It'd be like saying, I'm going to give our patients LSD and think that they're going to be more lucid after the surgery. But in fact, that's what was shown. And so we said, there's a low probability this is going to work, but if it did, this is a safe drug. We all have access to it. If it reduces delirium, it would be a huge win. And as we discussed even before we started the trial, even if it doesn't reduce delirium, if it doesn't change delirium, it will still be a win, because then everybody can feel comfortable using ketamine for analgesic purposes. And when we started this trial, we did a survey at 5 or 6 institutions that were participating, and most everybody thought that ketamine could reduce pain or spare opioid effect, but only a small percentage of people were using it because they were concerned about the psychoactive side effects. Now, by the time we finished the trial, the ketamine usage had tripled or quadrupled, not because of us, but because of changing times and recommendations that have been published. So we looked at delirium. We thought, even if it doesn't change, we know it's going to improve pain, so it will still be a win. And we also looked at depressive symptoms. Now, ketamine did not change the incidence of delirium. All right, no big surprise. It didn't reduce delirium. It's kind of a long shot. It was worth taking that long shot, but it didn't work. Now, on the other hand, it didn't increase the formal diagnosis of delirium. So we thought, okay, great news for the community, because now we'll show that the addition of ketamine has an analgesic property and reduces pain in the postoperative period. But then we had a surprise. Ketamine did not change pain scores compared to the placebo. Even on the day of surgery, so it's not even the delayed effect of post-op day 1 or 2 or 3, but even on the day of surgery, pain scores were not changed. So we said, okay, that's fine. If you look at the meta-analyses and different studies, ketamine can either reduce pain or pain could be the same, but you've reduced opioid consumption. And that would be a big win too, right? Especially in this day and age of an opioid epidemic. But then we found out ketamine didn't change opioid consumption. So there appeared to be no effect on delirium, no big surprise, but the big surprise was no change in pain scores, no change in opioid consumption. How could that be? This is in guidelines and recommendations. There's been a meta-analysis. But when we looked at the meta-analysis and we looked at the studies within the meta-analysis, we realized that our trial was 4 times larger than the largest trial in the meta-analysis. And what very commonly occurs in clinical research is when you do a small trial, you see a bigger effect. When you have a larger trial, more generalizable trial, such as a multi-center study, that effect tends to go away. And if you just add up a lot of small trials, it isn't really equivalent to doing a large pragmatic trial. We also found, and this is just in press in the British Journal of Anesthesia, that ketamine did not improve postoperative depressive symptoms, which was a little disappointing because there were some small studies that showed that it might be beneficial. And post-op depression is actually relatively common. It's about 10 to 15% of patients, and it can impact functional recovery. It's hard to do things when you're depressed. It's the very nature of depression. So ketamine didn't do any of the good things that we thought it was going to do, which I was not happy about. I did not go into this trial trying to disprove conventional wisdom about ketamine. I love the drug. It's fascinating, and it seems like it's effective. So then I was wondering, well, was the ketamine even work? Were we just giving saline to everyone? But then we found out ketamine did increase the incidence of nightmares and hallucinations. Not so much at a 0.5 mg per kg, but at 1 mg per kg. So it's just like when you grow up. Now I'm a parent. I've got 2 small children. I'm starting to discover that all the things that my parents told me were probably right, even though I rejected that for a long time. So all what we used to think, this is going to be more of a problem, it's going to add more problems than benefits, might actually be true. Now, a caveat, so we are more nuanced about this. Whenever you are interpreting a clinical trial, you don't want to either rush to implementation and you don't want to rush to de-implementation. We have to remember the specific conditions and methodology of the study. There are older patients. Maybe ketamine does work in younger patients. They got a single bolus dose. Now, some of the studies have shown infusions are effective. They got a bolus dose during the anesthetic. Other studies have shown that continuing an infusion in the postoperative setting can have analgesic benefit. And I'm certainly not saying that ketamine doesn't have analgesic properties, but these data suggest that the practice of a single shot of ketamine during the anesthetic might not have the benefit that we thought it had. And I used to do this. In fact, sometimes I still do it, and I know the data. I mean, that's the psychology of implementation science. So more work is going to be done. There are bigger trials. In fact, Paul Miles, who is a very famous trialist, is leading something called the ROCKET study, with about 5,000 patients looking at various outcomes related to ketamine. But what I can say from this, it's unlikely that a single dose of ketamine at the beginning of the case is going to have a profound effect on postoperative pain or opioid requirement. Now, the last part of the talk relates to this, the weirdness that can happen with ketamine. Now, I told you at the beginning of the talk that the original term for this was dissociative anesthesia, and this goes back to the mid-1960s. But why is it dissociative? That term's been thrown around, but nobody's ever really shown why ketamine induces dissociative effects. So, although this was an NIH-funded study about mechanisms of anesthesia, I kind of slipped in a psychedelic study within it. So we had 15 healthy volunteers. They received, again, this antidepressant dose of ketamine over 40 minutes. After they got it, we gave them an altered states questionnaire where we could formally assess some of the psychedelic and dissociative properties. And by the way, ketamine looks like a lot of classical or canonical psychedelics like LSD or psilocybin when it comes to certain experiences like the sense of transcending ego or boundlessness. Where it's really off the charts and unique is in this dissociative quality. And then we did both sensor and source level spectral analysis. So what does that mean? Well, one of the advantages of having 128 channels as opposed to 6 or 8 channels like the earlier studies is that instead of just looking at what's going on at the surface where the EEG is recording, you can actually model and try to figure out what the source of that electrical activity is. And what we found with ketamine is that it appeared to be preferentially suppressing alpha in this region of the brain called the temporoparietal junction. Now, if you were to take psilocybin, and I hope none of you are doing that right now. I know there's certain things that are legal in Colorado, but probably not that. You would see a reduction in alpha oscillations in your brain. And the more they were reduced, statistically speaking, the more of a psychedelic experience you'd be having. So alpha is thought to constrain neural dynamics, right? I mean, you got to get into the operating room. You got to walk, you got to be there on time. You can't be tripping out. You know, your brain can go in all sorts of different directions. You need to have a kind of a constraining function. And it's thought that alpha helps constrain those dynamics. When you take that away, as happens with LSD, psilocybin, and ketamine, you start to have a broader repertoire of neural connectivity patterns and a broader repertoire of experience. So we found something that's similar to other psychedelic drugs. But the temporal parietal junction is unique for another reason. It's thought to be an area in which there is the integration of different sensory data, and it's also an area that's known to be associated with a sense of body ownership. And in fact, when you have patients who have lesions in the area of the temporal parietal junction, I'm going to show you this in a minute, they have spontaneous out-of-body experiences. And this has been studied experimentally as well. So look at this area and look at where ketamine, again, the red means more, the blue means less, in terms of suppression of alpha. And look at the data from Olaf Blanke, who studied this extensively, of patients, and these are the different lesion points, of patients who have spontaneous out-of-body experiences. So it's a pretty remarkable similarity in terms of the right hemisphere temporoparietal junction, and raises the question, doesn't demonstrate anything, but it raises the question of whether ketamine's effects in the temporoparietal junction are driving this dissociative experience that's often reported by our patients. So to summarize these data, in humans, and in human surgical patients and healthy volunteers, ketamine anesthesia disrupts connectivity patterns in frontal parietal networks in a way that is qualitatively similar to the disruption induced by propofol and sevoflurane. And I should note, and this is important to note in this era of reproducibility crises, that this has been replicated by other labs, with other analytic techniques, with different recording techniques, with fMRI, magnetoencephalography. So it's a fairly robust finding. I don't know what it means fully, but it is a consistent finding. In rodents, anesthetic levels of ketamine depress connectivity, but they increase acetylcholine, especially once you start to get to sub-anesthetic levels. And we took advantage of that to try to re-engineer a typical slow wave-like anesthetic into something that incorporates a REM sleep component. And the sub-anesthetic ketamine during isoflurane anesthesia, paradoxically, increases anesthetic depth but accelerates recovery, possibly through enhanced cholinergic tone and enhanced communication across the cortex. In surgical patients in a multi-center randomized trial, ketamine does not appear to affect the incidence of delirium, reduce pain, spare opioid consumption, or reduce depressive symptoms, and especially at 1 mg per kg, ketamine increases the incidence of nightmares and hallucinations. And finally, in this last study I showed you in healthy volunteers, ketamine-induced alterations of consciousness are associated with reduced alpha power in temporal parietal junction. And that reduction of alpha power in posterior cortex is a common finding with other psychedelic drugs that have distinct molecular mechanisms, but that area of the brain thought to be important for sensory integration, body ownership, and I think a promising candidate for understanding why ketamine has this dissociative effect to begin with. So I'd like to thank the many people who actually do the work, and Phil Vlicides and Tarek Belbahar, who published some of the articles in Healthy Volunteers in the past few years. Dinesh Pal did the basic science. Stephanie and Anshul did some of that early human work in terms of the directed connectivity, other members of the lab from a few years back, and my close collaborator and friend Michael Avedon, with whom I conducted the podcast trial. I'd like to thank the NIH, funding sources for both conducting research and promoting research, and in particular, this grant was funded to study ketamine and nitrous oxide in non-human primates, and I didn't show you those data, and healthy volunteers. And I wanted to conclude with a quotation from Jamie Slay, who does neuroscience and is also interested in consciousness. And this comes from an editorial he wrote about one of our papers, and he said, arguably, the most important contribution the discipline of anesthesia can make to science will be in explaining the neurobiological mechanisms underlying the phenomenon of consciousness. I think we, as a field, have remarkable tools to modulate the nervous system, to strip away functions of the mind from functions of the brain, and ketamine in particular, because it has both psychedelic and anesthetic effects, can really give us some deeper insight into both the content of consciousness, what we experience, as well as the level of consciousness. Again, thank you for the kind invitation to speak. I thank you for your kind attention, and I'll be happy to address any questions you might have. Thank you. Yes, sir.
So given what you know, what is your practice with your surgical patients that you take care of as far as ketamine dosing and frequency?
So this gentleman asked me about what I do personally, what's my practice. So I'm a neuroanesthesiologist, so we have a lot of spine patients who come in, and the data do suggest that a ketamine infusion throughout the case can be helpful. I still often do deliver that infusion at the levels that have been reported and published, but I continue it throughout the case. It can also help enhance neurophysiologic monitoring. So again, I don't take the results of the trial and extrapolate to everything. It's always a mistake when you do that, either on the implementation or the de-implementation side. But what I don't do anymore is to just give a bolus at the beginning and think that that's going to have a great analgesic effect. Yes, sir.
Great talk. I really appreciate you doing that for everybody here does. Question. You talked about ketamine. The ketamines, can you talk a little more about, it's really a mixture of two drugs, RN and S, and even maybe their specific metabolites may be interesting in a differential effect?
Yeah, so great question. He was asking about the different forms of ketamine. We haven't really studied that in detail. I should know more about it than I actually do, but your point is very well taken in terms of trying to understand which form of ketamine is having which effect in which part of the brain. And so I think that's very important for ongoing research. Vesna. So the question related to chronic pain patients, and that's where, again, the data from this trial don't necessarily speak specifically to that. We would need another trial or a bigger trial where we could have a subgroup analysis to see, well, are there certain populations for whom ketamine can be effective? We didn't really pull out patients who had chronic pain or who were opioid dependent to see whether or not they would have a particular benefit. So again, that's why I'm being cautious and nuanced and not saying don't give ketamine anymore, but rather just be thoughtful about the way that you're giving it and to the patients to whom you're giving it in light of recent data.
You mentioned that others hypothesized that they had an anti-inflammatory effect, the ketamine. With your cannula data, you believe it is anti-inflammatory?
So the question related to the anti-inflammatory effects, we actually haven't studied that directly, but I think there have been meta-analyses and there's some general agreement that ketamine does in fact have anti-inflammatory effects. Whether or not that has some sort of functional effect in the brain, I think is unknown. The one trial I cited by Judith Huditz, and actually her husband Tony Huditz works with us in our department, demonstrated that the reduction in delirium and postoperative cognitive function was associated with a reduction in inflammatory markers. But I haven't done anything definitively with that topic. Okay, well, thank you very much, and maybe I'll see some of you tomorrow.